Y'all are making me so nervous over there. Okay. So we're going to talk really quickly through transcription because we talked about it earlier. But I want to refresh and make sure that everyone feels good about it. So this is, remember, purple's representing DNA, and then the green is representing RNA. Does anybody remember what type of RNA? mRNA, messenger RNA, right? Okay. And remember that all we're doing is transcription is going from DNA to now we have mRNA, which matches up with the DNA. Are we transcribing like the whole chain of DNA at once?
Yeah.
No.
No, we're doing portions.
What's that portion called?
The Asian guy's a gene.
A gene. Good. That's DNA replication.
Yeah,
That's DNA replication. So we're doing transcription, which is going to be taking a gene of DNA, which is just like a section of DNA that codes for a particular protein, and we're just transcribing that section because that's the only protein that we want to make. Okay, so this purple represents the DNA, and then the green represents the mRNA. So we have the DNA, and RNA polymerase comes and it unwinds and unzips it. Now we're kind of looking at two different strands that one is going in a 5 to 3 direction, one's in a 3 to 5. We already know that about DNA. Okay, so what we're going to do is we're going to kind of look at the gene as a section now, and genes are not 4 codons long, or sorry, 4 nucleotides long. They're going to be like thousands of nucleotides long, but just for this picture's sake, this is a gene. Okay, so the promoter region, that's going to be where transcription starts, and the terminator region, that's basically the end of the gene. That's where transcription needs to end. So there are going to be mRNA nucleotides, free nucleotides, just kind of out and around. And what's going to happen is that RNA polymerase brings over the mRNA nucleotides, starting at the 5' end of the mRNA and moving the way up to the 3' end of the mRNA. So we brought over a G, we brought over a C, and then to match up with the adenine, we had to bring over a U of mRNA. And then to match up with the thymine, what did we need to bring over? Adenine. Good. So now we have our matching sequence of mRNA. Yep. Mm-hmm.
So all of those that you added on were from where? Like where?
They're free nucleotides, so they're not attached to anything. They're like a phosphate, a ribose sugar, and a nitrogenous base that aren't bonded to anything yet.
Okay.
Did that answer your question? Okay. So once we've made it to the terminator region, then transcription is going to stop, and this mRNA sequence is going to break off and go to the cytoplasm where the ribosomes are, and that's where translation is going to take place. So transcription, we start off with DNA, and we're going to make an mRNA code that matches up with the DNA. That mRNA code is going to travel out of the nucleus, go to the cytoplasm to the ribosome, and go through a process called translation where we actually make the protein. Okay? So the DNA technically codes for the protein, but the mRNA is kind of like the go-between. Okay?
So when is it made into mRNA? Right there?
Yes, this is mRNA right here.
Once they're added?
Yeah, so these nucleotides that are matching up with the DNA is called mRNA. And then it breaks off and leaves, and the DNA comes back together and rewinds.
Okay.
Okay?
So when do you know?